Hi Ninja Nerds, in this video we are going to talk about adrenergic receptors. These are extremely important, so let's go ahead and get started. So adrenergic receptors, we're going to talk about these in two classes, right? So here, let me write up here, we're going to have adrenergic receptors, okay? Adrenergic, meaning that they're very, very sensitive to what? Adrenaline. But again, we call adrenaline, nowadays we actually call it epinephrine, but there's also other neurotransmitters, such as norepinephrine. So when we talk about adrenergic receptors, we're going to talk about specifically the receptors that are sensitive to epinephrine and norepinephrine. Okay, and there is other different types of molecules that are very similar in their activity. We call those adrenergic agonists. Those would be things like isoprenaline and dobutamine, all those different types of things. We'll talk about those very, very briefly. But what I want you to understand is that adrenergic receptors are sensitive to chemicals such as epinephrine and norepinephrine. Now, next thing I want to do is I want to say, okay, let's separate these two receptors. Okay, well, let's separate these receptors into two subclasses, right? So now we have two classes here. We're going to have alpha adrenergic receptors. So we're going to put this as alpha adrenergic receptors. And the other one is going to be beta adrenergic receptors. Okay? Now, what is important to know about these is specifically their subtypes. If we really want to dig into this and really understand it, we have to understand their subtypes. So alpha adrenergic receptors, there's actually two types. Alpha 1 adrenergic receptors and alpha 2 adrenergic receptors. Okay? Beta adrenergic receptors, there's three types. Okay? There's three types, and these are simple. Beta 1 adrenergic receptors, beta 2 adrenergic receptors, and beta 3 adrenergic receptors. Okay, so now we know what adrenergic receptors are. The receptors that are sensitive to epinephrine, norepinephrine. We know there's two classes, alpha and beta. We know that alpha is subclassified into alpha 1, alpha 2. And beta is subclassified into beta 1, beta 2, beta 3. Why should we know this? Now, here's what we're going to talk about with respect to this cell. Each one of these receptors, for the most part, respond to a specific chemical, such as epinephrine, norepinephrine, but exert an intracellular signal that is unique and different. So what do I mean? Alpha 1, we're going to go over it, but alpha 1 adrenergic receptors primarily work through GQ proteins. So GQ coupled protein receptors, okay? Alpha 2 is primarily through G inhibitory, okay? Primarily through G inhibitory. But beta 1, beta 2, and beta 3 primarily work through what's called G stimulatory. So now, what I'm going to do is I'm going to go over the different intracellular mechanisms of these adrenergic receptors in detail and why that's important to know. Okay, so let's go ahead and do that then. Okay, first things first, let's start off with the alpha 1 adrenergic receptor. All right, so let's say here we're going to have this part of the cell membrane, right? And at this part of the cell membrane, here's going to be our receptor. And this receptor is going to be an alpha 1. We're going to start with that one first. So here's going to be our alpha 1 adrenergic receptor. Okay, so alpha 1 adrenergic receptors, we said, act through GQ protein coupled receptors. So let's pretend here that we have either one of the two chemicals. We can have norepinephrine or we can have epinephrine. Both of these guys can act on the alpha 1 adrenergic receptors. Now, technically, if we want to be particular, epinephrine has a higher affinity for the alpha 1 than norepinephrine does. Anyway, both of these guys can come over here and act on this receptor. Now, let's say that they bind to the receptor. We know that these receptors are usually linked or connected with a G protein. We said in this case that the G protein is actually GQ. GQ protein. We said that GQ is normally bound to GDP, but it throws away the GDP and takes on GTP. GTP, when it binds to the GQ protein, stimulates it. Then this GQ protein will come and stimulate an effector enzyme that's located inside of the plasma membrane. This enzyme is what we refer to as phospholipase C. So we call this enzyme here phospholipase C. What phospholipase C does is it breaks down a chemical, which is called phosphatidyl inositol diphosphate. And what it does is it breaks it down into two chemicals. One is called diacylglycerol. The other chemical is called inositol triphosphate. Now, diacylglycerol goes and activates a special type of enzyme. We call this enzyme a protein kinase. And if we really want to be particular, protein kinase C. So this is going to stimulate that. IP3 works through a different mechanism. It actually activates different types of calcium storage centers. And when it activates these calcium storage centers, like the sarcoplasmic reticulum or the endoplasmic reticulum, it increases the release of calcium out of those structures. Now, calcium will then go and bind on to a special protein that's usually located within these cells, which is called calmodulin. Now, when calcium binds with calmodulin, it forms what's called a calcium-calmodulin complex, or sometimes they even say kinase. Okay? Now, why is this important? Okay. Calcium-calmodulin complex and protein kinase C can phosphorylate different types of proteins. So this can come over here, and what it can do is it can phosphorylate different types of proteins. Let's say here I draw in red, this is going to be some type of protein here. And here's its amine end, and then here is its carboxyl end, right? What it's going to do is it's going to put phosphates onto this protein. And when you phosphorylate proteins, it can either activate or inactivate the enzyme. It all obviously depends on that protein or enzyme in this case. Same thing with the protein kinase C. Just remember, it can exert the same type of activity here by phosphorylating different types of proteins / enzymes. And why is this important? Depending upon the tissue, these proteins might be different types of membrane proteins. And maybe this membrane protein is some type of channel. And it can allow for cations to leak into the cell. Maybe these proteins can actually go and do other different things, like certain metabolic functions. Or maybe it could actually go to the nucleus and trigger DNA transcription. So there's many different functions of these different types of proteins or enzymes. So it can change the membrane permeability. There's so many different functions, okay? And again, that's not the biggest thing. The biggest point here is I want you to understand that the intracellular mechanism is increasing calcium and protein kinase C, phosphorylating proteins that can have various different functions within inside of this cell. That's alpha 1. Alpha 2 is a lot easier, okay? So now let's do alpha 2 here. Let's do this one here in a nice purple color. So here, we're going to have a receptor right here. And alpha 2 is thankfully a lot easier here. So here's our alpha 2. And alpha 2 receptors, alpha 2 adrenergic receptors, are actually going to be G inhibitory, we said. So whenever epinephrine or norepinephrine bind onto these receptors, and again, epinephrine has a higher affinity than norepinephrine, it'll activate an intracellular G inhibitory protein. Now, what's important about this is that the G inhibitory protein has 3 subunits: an alpha inhibitory, a beta inhibitory, and a gamma inhibitory. Okay, now, why is this important? Whenever epinephrine binds onto the adrenergic receptor, it separates these two. So alpha inhibitory separate and beta inhibitory separate. Now, the alpha inhibitory come over here, and they bind onto this special type of membrane protein here, this special type of effector enzyme, and we're going to call this adenylyl cyclase, right? So adenylyl cyclase. You might see in other videos I've usually been lazy and just denoted as AC, right? All right, but anyway, this alpha inhibitory subunit is going to come over here. And what it's going to do is it's going to inhibit the adenylyl cyclase. Now, adenylyl cyclase is normally responsible for being able to convert ATP into a molecule called cyclic AMP, right? But if the alpha inhibitory unit is binding to the adenylyl cyclase, it's inhibiting it. Is it going to be able to make cyclic AMP? No. So as a result, the cyclic AMP production will decrease. Cyclic AMP is normally supposed to activate protein kinase A, right? And then protein kinase A is important because it can go and do what? Phosphorylate different proteins and enzymes. But if it's not able to do that, will it be able to phosphorylate the different proteins and enzymes? No. So that's the whole significance here. Now, what else? What about this beta and gamma inhibitory unit? Let's do this in orange. Now, there's special channels, usually in most of these cells that have this G inhibitory unit, and these are sensitive to the beta and gamma subunit. So let's say that the beta and gamma subunit come over here to this orange channel, and it binds onto this channel. When it does that, it opens this channel up, right? So it opens this channel up and allows for potassium ions to leave the cell. If potassium ions leave the cell, what's that going to do to the inside of the cell? It's going to make it negative. It's going to try to hyperpolarize the cell. You're going to see why this is important, because the main location of alpha-2 adrenergic receptors is on the presynaptic nerve terminals, and it's going to make sense when we go over that. Okay? Again, we're just laying down the groundwork for this stuff. Now, easiest thing is that's alpha-1, alpha-2. Beta-1, beta-2, beta-3 all have the same intracellular pathway. And thank goodness for that. What is that one? That one is G-stimulatory. So now we've already pretty much laid down the groundwork. So watch this. Okay, here we're going to have our next receptor. And again, this can be beta-1. It can be beta-2. Or, here, let's do it like this. Beta-1, beta-2, or beta-3 adrenergic receptors. These guys, again, they're sensitive to epinephrine and they're sensitive to norepinephrine. When they bind onto this receptor, they're going to activate an intracellular G-stimulatory protein. That G-stimulatory protein is going to pop off a GDP and pick up a GTP, activating this enzyme. If this is stimulated, what is it going to do? It's going to act on adenylyl cyclase. It's going to stimulate adenylyl cyclase. What is that going to do? That's going to increase the conversion of ATP to cyclic AMP. That's going to increase the actual activation of protein kinase A, which is then going to go and phosphorylate different types of proteins and enzymes within inside of the cell. So now, we should understand the different types of receptors. We should understand their difference in intracellular pathways and the overall significance of it. Now what we got to do is we got to go ahead and talk at all the different types of target organs or locations of these different types of receptors. So let's go ahead and do specifically focusing on the blood flow. I think that's a good way to get started on these because it kind of lays down a nice groundwork for us. So first thing we're going to talk about is these adrenergic receptors located on the tunica media, right? You know, within blood vessels you have, here, a really simple diagram here. Let's say here's a blood vessel. You have an inner lining, which is called the tunica, intima. After that, you're going to have smooth muscle lining, which is called your tunica media. And then after that, you have some connective tissue outside of it, which is called the tunica externa or the tunica adventitia. The tunica media has a lot of adrenergic receptors. And because of that, it's very, very sensitive to epinephrine, norepinephrine, so it can control the diameter of the blood vessels. So that's where we're going to start with first, talking about the adrenergic receptors on these tunica media, these blood vessels. So first one we're going to talk about is the blood flow to the integumentary system or the skin. Okay, I want you to think about these things logically. It's best when we think logically. If you're in a sympathetic crisis, we know that the sympathetic nervous system primarily functions in different types of fight or flight or crises-like type of situations. So we've used the example of like running away from an orangutan or fighting with Chuck Liddell, whatever. Something that's going to get you hyped up and really, really nervous, okay? Now think about this. The skin, is that going to help you run away from orangutan or fight Chuck Liddell? No, it's not going to do anything for you. So we don't really care about the blood flow going to the skin. We'd rather that blood go to the skeletal muscle so that we can run or fight. So in that situation, I want you to think about that. Usually, on the vascular smooth muscle of the skin is going to be what's called alpha 1 adrenergic receptors. That's what we're going to find there. Now think about it. Alpha 1, what did it do before? Well, it increased the GQ, which increased the calcium and protein kinase C. Oh, I know that calcium helps to activate the muscles to start undergoing contraction. They need that for the cross bridge formation. Oh, that's why. Okay, that makes sense. Now, the blood flow to the skin is going to be reduced. If the blood flow to the skin is reduced, what normally will someone in like a sympathetic situation, what color will their skin be? Usually they're going to have more of like a palish skin appearance or skin hue, right? Okay, so that's one big thing. And the next thing we should understand, you know there's a muscle that's actually connected to the hair follicle? And whenever we're in a sympathetic situation, like we're scared, usually the hairs stand up, okay? Well, there's a muscle there called the erector pili muscle. And that erector pili muscle, guess what type of receptor is there? An alpha 1 adrenergic receptor. And again, what is that going to do? That's going to cause the contraction of this actual muscle, which is going to cause the hairs to stand up. So as a result, when this is stimulated, the erector pili muscle, when that's stimulated, it's going to cause the hair to stand up. Okay. So far we got that. Okay, so that's the skin. So the first thing we want to make sure that we do is we divert the blood away from the skin, vasoconstricting the blood vessel. If we do that, the skin will be more palish, and also it's sensitive to the erector pili, has alpha 1 adrenergic receptors. If it contracts, it causes the hairs to stand up. Next place is going to be going to your kidneys. So now let's think about the kidneys or the renal system. So this is going to be specifically for the kidneys. The blood flow going directly to the kidneys. So here's our kidneys here, right? So kidneys and the kidney, right? We specifically want to know what's going to happen to the blood flow in the kidneys. Well, if you're running away from an orangutan or fighting Chuck Liddell, you don't want to pee yourself. So you want to be able to restrict the urine from being formed. So if that's the case, do I want a lot of blood going into the kidney so that they can filter that blood and then make urine? No. So if that's the case, I'm going to want to reduce the blood flow to the kidneys. If I want to reduce the blood flow to the kidneys, what type of receptors would you expect to be on these blood vessels going to it? It would be alpha 1 adrenergic receptors. And again, that's going to reduce the blood flow, BF for blood flow, right? And then as a result, you're going to make less urine as a result of that, okay? All right, so that covers that. Next one, the GIT. If you're running away from an orangutan or fighting Chuck Liddell, are you going to be worried about digesting like a hot dog or something? No, you don't give a rip. You don't care about that right now. You'd rather spend that blood going to your muscles or useful vital organs. So if that's the case, let's constrict these puppies here. So now what we're going to do here is for this one, the blood flow going to the GIT, our splanchnic circulation, We don't really care about that. So let's constrict those bad boys. So we're going to have the alpha 1 adrenergic receptors located right here. And if they're located right there, it's going to reduce the blood flow to the GIT, decreasing different digestion, absorption, decreasing secretions, right? So that's the purpose there. Okay, next one. This one's a little interesting. Okay, so far we've been pretty much hitting all the unnecessary or not vital organs, like the skin, the kidneys, the GIT. What about the blood flow to the skeletal muscles? Okay, well, I know that if I have a lot of blood flow going to the skeletal muscles, that's going to be helpful for me because I got to run away from this orangutan or fight Chuck Liddell. So I need a lot of blood flow going to the muscles. But if I constrict them, if I constrict these blood vessels, that's not going to help me at all. Instead, I need to dilate them. So here's what I want you to remember. If that's the case, then, if we want to dilate these bad boys, the main receptor that's located on these guys is going to be what's called beta 2 adrenergic receptors. Because the beta 2 adrenergic receptors, they work through a G-stimulatory pathway, right? So they're going to increase cyclic AMP. If they increase the cyclic AMP, it's going to increase the protein kinase A, right? But what protein kinase A does is it actually inhibits a special enzyme that's controlling the blood flow to these actual muscles. So in that case, the muscles are going to relax. So there's going to be increased perfusion to the muscles. So what should happen to the blood flow to the muscles? The blood flow to the muscles should be increased. And these blood vessels should dilate, okay? So the skin constricted, the kidneys constricted, the GIT constricted, but the blood flow to the skeletal muscles vasodilated, increasing the blood flow. All right, what about the blood to the actual heart itself? You know, the blood has a system called the coronary circulation that serves itself. Here's what's interesting. This one has kind of about an equal amount of alpha 1 and beta 2. Here's what I want you to remember. These receptors, alpha 1 and beta 2, these are usually the only ones specifically that you're going to be found on our vascular smooth muscle. So with the receptors that are present on the tunica media, there is mainly of two types, alpha 1, beta 2. Alpha 1 is going to cause constriction. Beta 2 is going to cause relaxation. This one has about an equal amount of beta 2 adrenergic receptors and alpha 1 adrenergic receptors. Now, why is that significant? Our heart cannot rely upon the sympathetic nervous system or the autonomic nervous system because it's fluctuating all the time. So if the blood flow going to our actual myocardium is actually dependent on the autonomic nervous system, it could be very, very dangerous. So because of that, the blood flow going to the heart itself is under what's called metabolic control. In other words, if there's a decrease in oxygen, right? If there's some type of situation like a decrease in oxygen or an increase in what's called adenosine, these type of things can actually dilate the blood vessels. So because of that, they have what's called an autoregulation. In other words, they can regulate their blood flow on their own based upon different types of metabolic substances. So if there's like a decrease in oxygen, increase in adenosine, that could actually cause the blood vessels to dilate, increasing the blood flow. So depending upon the chemical factors that are in the blood, that can alter the actual blood vessel vasoconstriction and dilation. So I want you to remember that. So the autonomic nervous system doesn't have a very significant effect on the coronary circulation. In the same sense, central nervous system. We can't leave the blood flow to the central nervous system or to the heart at the mercy of the sympathetic nervous system because it fluctuates so much. So if that's the case, then we do not want to just have primarily alpha 1 adrenergic receptors. We want to have a mixture of alpha 1 and beta 2 adrenergic receptors. And again, If that's the case, then the actual central nervous system has to have its own regulating system. And again, we call that autoregulation. And this autoregulation is usually through what's called a myogenic mechanism. And we've talked about this in many other videos, like the microcirculation video. Okay, but the myogenic mechanism is just basically if there's an increase in pressure, it constricts the vessel. If there's a decrease in pressure, it dilates the vessel. So this has its own way of being able to control the blood flow that's not dependent upon the autonomic nervous system. Okay, sweet deal. Okay, now that we covered that, let's kind of start working our way down through multiple different types of structures. Okay, so if we get to the eyeball, we've talked about this again, and a lot of this is going to be kind of like a continuation of what we talked about in our sympathetic nervous system video. We're kind of just going through the target organs that we had there and talking about the receptors that are present there. Okay, so if we think about this, the sympathetic nervous system wants us to see far. If you guys remember, we said we wanted far vision. So there was two different muscles that we wanted to target here. One was called the ciliaris muscle. If you remember, we have the ciliaris muscle. And the ciliaris muscle, we said, should specifically relax. If this muscle relaxes, then what happens is these little zonules, the suspensory ligaments or the ciliary zonules, those will become nice and tight. If those become really, really tight, the lens gets really flat. So if that's the case, then we don't want this muscle to contract, because if it contracts, it pulls the muscle up this way and causes those zonules to get really, really relaxed. So instead, we want this muscle to relax. If we want it to relax, what type of receptors would you expect to be there? Beta 2. So beta 2 adrenergic receptors should be located there, okay? And again, because of that, the ciliaris muscle, it will relax. If the ciliaris muscle relaxes, this is going to cause the lens to flatten. And if the lens flattens, that's going to allow for the vision to be set for far away objects. Okay, next thing. What about this muscle? You know the muscle around the iris, you have specifically, they call it the dilator pupillae, right? This muscle, if it contracts, it opens up the pupil hole. So if that happens, it causes pupillary dilation. So now, as a result of that, it's going to cause pupil dilation. And again, if we dilate the pupils, it's going to increase the amount of light rays hitting the retina. And again, both of these are going to function for overall far vision. And it should make sense. If you're running away from an orangutan, you want to know all the different types of options, that you can go to, all right, to be able to escape that. All right, sweet. Now, here's the thing, though. We said that there's beta 2 adrenergic receptors in the ciliaris muscle. But we said that the dilator pupillae, its function is to open the pupil. Well, that means that the muscle contracted. If the muscle contracted, then it has to be an alpha 1 adrenergic receptor. Okay? Alpha 1 adrenergic receptor. Okay. Now let's go to this one, the salivary glands. Not super, super crucial to this, but again, it's understanding why, what would happen here. So if we go to the salivary glands, are you going to be worried about making a lot of saliva, being able to digest different carbohydrates or maybe lipids within the mouth? No. So you're not really obsessed with that. So because of that, the salivary glands have specific receptors on them. And generally these are going to be beta 2 adrenergic receptors. And what this is going to do is it switches the salivary secretions into making less water and electrolyte rich saliva. So it's going to decrease the amount of water and electrolytes that are in the saliva and it's going to increase the mucin proteins and increase different types of salivary enzymes. If that's the case, then it makes the overall salivary secretion, it makes it a thicker saliva, okay? So it makes the saliva really thick and really viscous. Another thing is there's blood vessels that are going to the actual salivary glands. And if the blood vessels are going to the salivary glands, the actual receptors can be present on these actual blood vessels going there. And if that's the case, we can constrict these blood vessels and decrease the blood flow to the actual salivary glands, which would decrease the water and the electrolytes that we can filter out. So if that's the case, we're going to want the blood vessel to constrict. So this should be alpha-1 adrenergic receptors. And again, that's going to decrease the blood flow, which is going to decrease the amount of water and electrolytes that are going to be put into the saliva. Okay? All right. Now let's go over here. We already talked about the blood flow to the central nervous system, but we have to talk about one more thing. Okay, we said that we have these things called presynaptic nerve terminals, right? So here, let me actually fix this for a second. And let's do this like this. So here you're going to have your preganglionic motor neuron, right? Coming out of the thoracic region of the spinal cord. You know the thoracic region of the spinal cord is from T1 all the way to about L2, right? So we have T1 all the way to L2. These are your preganglionic motor neurons, right? But let's say that just in general, we come over here and we have, like, let's just say that this is a ganglion. And then here, I'm going to balloon up this axon terminal, okay? If you guys remember, we've talked about this in certain videos, that you know that we have vesicles, synaptic vesicles that are located within the axon nerve terminal. And they consist of neurotransmitters. In this situation, we're going to say that it's norepinephrine. So in this one is norepinephrine, norepinephrine, norepinephrine. And we know that whenever there's an action potential being carried down the axon, it opens up special voltage-gated calcium channels that are located within the axon terminal. So let's say that here in green is going to be these voltage-gated calcium channels. And we said that whenever the action potential travels down, it opens up these calcium channels, calcium rushes in, and then stimulates the migration of these vesicles to the actual membrane where it will fuse. And if it fuses, it will release these chemicals, this norepinephrine, by exocytosis. Well, guess what? There's another receptor that's kind of sitting right here, okay? Let's put him right here. Let's make it nice and red here. Here's this receptor. Here's a nice little receptor here. And this receptor is going to be very sensitive to the norepinephrine that's being released. When norepinephrine is released, what it can do is it can go and act on its target organ. Let's say, just say here's its target organ, right? But another thing that can happen is that norepinephrine can come over here and act on these receptors that are present on the actual presynaptic nerve terminal. These receptors are called alpha-2 adrenergic receptors. If alpha-2 adrenergic receptors are activated by this norepinephrine, guess what they're going to do? They're going to decrease the actual cyclic AMP production, which is going to decrease the protein kinase A. If protein kinase A is actually in decreased amount, guess what can happen? It can actually inhibit these calcium channels from opening. Another thing is, if it binds onto the alpha-2 adrenergic receptors, remember what else it did? It opened up special channels on the membrane that were sensitive to the beta and gamma subunit and allowed for potassium to leak out of the cell. If potassium starts leaking out of the cell, it makes the cell increasingly more negative. If the cell becomes increasingly more negative, is it going to be able to be stimulated? Is it going to be able to depolarize? Open up the calcium channels that calcium can come in and cause the release? No. So because this norepinephrine binds onto the alpha-2 adrenergic receptors, it can cause potassium ions to leave, hyperpolarizing the cell, making it unable to release norepinephrine. So that's a cool little mechanism right there. Okay, so that's what I want you to remember. The biggest target organ of alpha-2 adrenergic receptors is these actual nerve terminals, the presynaptic nerve terminals. And what norepinephrine can do is actually bind onto them, increase the potassium ion, efflux, hyperpolarize this nerve terminal, and make it unable to allow for calcium to come in. If calcium cannot come in, it's not going to trigger the release of norepinephrine. That's so cool. All right, that covers that part. Now, let's go and let's talk about the heart. The heart, we already know the blood flow. We know that it has its own autoregulation, and we know that the central nervous system has its own autoregulation mechanisms. This one more by metabolic, the central nervous system more by myogenic. But the heart has its own nodal system, if you will. Let's do this in orange here. It has its own nodal system, okay? Like the SA node. If you remember, it has the AV node, it has the bundle of His, and it has the bundle branches, and the Purkinje system. Well, on the SA node and the AV node and the bundle of His, they have special receptors. And these special receptors that are located on the SA node, the AV node, the bundle of His, the bundle branches, but primarily SA node, AV node, and the bundle of His, these are specifically going to be beta 1 adrenergic receptors. Easiest way to remember is beta 1 adrenergic receptors are usually only located in about 2 important places. One is the SA node, specifically the heart, the myocardium of the heart, SA node, AV node, bundle of His, and another part which is the actual contractile myocardium. And the second place is the juxtaglomerular cells in the kidney that cause the release of renin. And there's one more which is, it's not significant, but it can actually cause the release of ghrelin from the stomach, a hunger hormone. But the big ones is beta 1 are located on the heart and the kidney. So remember that. 2 big ones, and you covered beta 1. Now, we said that it's located on the SA node, AV node, bundle of His, and the actual contractile myocardium. So if that's the case, then if it's located on all these different locations, I want you to remember what's the overall effect. If it acts on the AV node, SA node, and the bundle of His, it's going to increase the action potentials. That is going to increase the heart rate. Heart rate increases cardiac output. Increasing cardiac output increases blood pressure. So that should make sense. Okay? At the other end of this, it's going to increase the contractility, increasing the calcium loading into these cells. If that happens, it's going to increase stroke volume. If you increase stroke volume, increase cardiac output. And if you increase cardiac output, you increase blood pressure. Okay, so we know that there's beta 1 adrenergic receptors located on the SA node, AV node, bundle of His, and the contractile myocardium. All right. Let's go to the next one, which is the muscle. So we talked about the blood flow, but there's something else really special about the muscles. There's actually these things called muscle spindles. And these muscle spindles are actually responsible for being able to determine the stretch. They're proprioceptors, basically. They help us to determine the position of our actual muscles in space, and specifically determining the stretch of the muscles, right? Either the beginning of the stretch or the progression of the stretch. Well, they have special receptors on them. And these receptors that are present on them can increase the muscle spindle firing. If you increase the muscle spindle firing, it increases the contraction of those muscles, which causes a slight tremor effect. That's why sometimes, if any of you, I have asthma, so sometimes I have to take an inhaler, right? Albuterol. And albuterol is just basically a beta 2 agonist, meaning it acts on beta 2 receptors, just like epinephrine and norepinephrine would. When it binds onto it, its main goal is to cause dilation of the actual bronchi. That's the whole purpose of albuterol. But as a side, it also can act on these muscle spindles. And if any of you do have asthma and you've taken albuterol, sometimes you're a little shaky. And it's a little bit of a tremor. And that's because of these actual muscle spindles. So there's going to be beta 2 adrenergic receptors on the muscle spindles, which is going to produce kind of like a little tremor. Okay? It also has another function too, that with the beta 2 adrenergic receptors, our muscles are constantly depolarizing and repolarizing. And so what they're doing is they're pushing out potassium and they're constantly bringing in sodium. And because of that, we want to be able to maintain that gradient. So there's a protein called a sodium potassium ATPase. And what it does is it pumps 3 sodium out of the cell and 2 potassium into the cell. Well, the beta 2 adrenergic receptors, guess what they can do? They can stimulate this dang marker. It can stimulate these sodium potassium ATPases and increase the influx of potassium and increase the efflux of sodium, maintaining those gradients. So that whenever there's another stimulus to the muscle, it'll be ready to contract. It'll have the necessary ions in the extracellular fluid and in the intracellular fluid. So it'll be able to depolarize and repolarize. Okay, so we covered that. So that's going to be important for the sodium potassium ATPases and the muscle spindles. Let's go up and we go to the GI tract. The GI tract, we already know the blood flow. We don't really care about the blood flow, so we want to constrict the vessels going to the GIT. But another thing I want you to remember is, are you going to be worried about contracting? You know, there's receptors here, right? You have smooth muscle all around this actual GIT, right, within the muscularis externa. You don't want that to contract. You don't want to waste energy, because remember, the sympathetic is for fight or flight. Parasympathetic is for resting, digesting, urinating, defecating. We're not going to want to enhance the GI motility. So if that's the case, we want to have inhibitory receptors, which is our beta 2 adrenergic receptors. And if we have these on all of the GI smooth muscles, throughout the entire GI smooth muscle, what is it going to do? It's going to decrease GI motility and decrease GI secretions. And as a result, it'll decrease the absorption across the GI tract. So remember that there's beta 2 adrenergic receptors on the actual smooth muscle of the GI tract. But here's where it gets a little tricky, okay? It gets a little tricky because there's sphincters of the GI tract. One is called the pyloric sphincter. So right here we have what's called the pyloric sphincter. And then there's another one right here which protects your doo-doo hole, and that's called the internal anal sphincter. Your sympathetic nervous system is very, very kind to you. He prevents you from crapping your pants, right? So nicely, he has to have receptors here that are stimulatory and will constrict. So in this one, the pyloric sphincter, it constricts because it wants to prevent the movement of chyme out of the stomach and into the small intestine. That's the purpose here for the pyloric sphincter. We want this to contract so that we can prevent the movement of the food. We don't want the food to go from the stomach into the small intestine. Next thing is we're not really worried about dropping a Lincoln log, okay? So because of that, we don't need this internal anal sphincter to contract. We don't need it to relax because we don't want to allow for the poo to move down. So if that's the case, we want this sucker to be constricted hard, right? So you want there to be alpha-1 adrenergic receptors there because it'll constrict it and prevent that log from going down. Okay, let's move on next to the actual kidney. All right, so we did the kidney, we did the blood flow. But remember I told you inside of the kidney is a special apparatus. We talked about this in the renal system, so there's no use in going into super detail here. But if you remember, we had these structures which are called your JG cells. JG cells. And these guys were really specific. They were baroreceptors, and they could respond to their own blood flow by releasing renin. Well, at the same time, they also have receptors on them that are sensitive to epinephrine and norepinephrine. And what did I tell you? Beta 1 is only located on real two important places, the heart and the JG cells of the kidney. So right here, we're going to have beta 1 adrenergic receptors on the JG cells. And again, what is the significance of it? When epinephrine and norepinephrine bind here, the whole significance is it's going to stimulate the release of renin. And if you remember the purpose of renin cleaved angiotensinogen that was made by the liver. Angiotensinogen converted into angiotensin 1. Angiotensin 1 went to the lungs, got converted into angiotensin 2 by ACE. Angiotensin 2 then bind onto the actual smooth muscle of our vascular smooth muscle and constrict those puppies. And then on top of that, it released ADH from the posterior pituitary and aldosterone from our adrenal cortex to increase the sodium and the water reabsorption from the kidneys, to increase our blood volume and our blood pressure. So it should all make sense. Okay, now let's think about this again. Here's our ureters, right? Are you going to be worried about spending and wasting energy and contracting the ureters to push urine down? No. Just like you didn't want to drop a Lincoln log, you don't want to pee your pants. So because of that, you want to minimize the urine output, right? That's the whole goal here. You want to try to minimize that urine output. That's for the parasympathetic nervous system. So if that's the case, then are you going to want to have these ureters contract? No, you're going to want them to relax. So smack on one that's going to do that. That would be the beta 2. The beta 2 adrenergic receptors are going to cause the smooth muscle in this area to relax. And if the ureters relax, is it going to push urine down? No. Now, obviously, urine can move down by gravity, so there's not a significant effect here, but the ureters do have a little bit of peristaltic activity that they can push urine down. Next thing, your bladder. Are you going to be worried about evacuating the bladder, causing the bladder to contract? No, you don't want to urinate. So because of that, you're going to want to smack your beta 2 adrenergic receptors on this puppy. And recent research has also showed that there's actually beta 3 adrenergic receptors present on this smooth muscle here, which is, they call it the detrusor muscle, okay? But big one is the beta 2. But again, just want to show you that there is another one out there, that being beta 3. And again, this is going to relax the bladder, and if it relaxes, it's not going to cause the evacuation of the urine into the urethra. Next thing, we have another protective mechanism. So not only have we decreased the urine flow through the ureters, we've also decreased the blood flow to the kidneys. We've also decreased the contractions of the actual bladder. Let's squeeze this sucker as hard as we possibly can, right? So let's go ahead and have this internal urethral sphincter also really, really tight. So in the same way that our internal anal sphincter was constricted, we're going to want this internal urethral sphincter constricted. So let's throw on an alpha 1 adrenergic receptor. Okay, now let's come up here to the actual adipose tissue. So the adipose tissue, why would this be important? Well, you know, your adipose tissue, your sympathetic nervous system is trying to get as much nutrients, as much blood flow to your vital organs as possible, because we need those muscles to have as much ATP as they can to contract and to perform the fight or flight activity. These are stored with energy systems called triglycerides, lipids. If our sympathetic nervous system can tap into that and get some of those lipids broken down, we now have a very, very powerful energy source. So now our sympathetic nervous system should have receptors on our adipose tissue. So this is our adipose tissue, or our adipocytes, right? And what happens is there should be receptors here which are going to respond to the epinephrine and norepinephrine and break down. Look, here's our glycerol, here's our fatty acids. We've drawn this in many, many diagrams before. This is the glycerol and the fatty acid, glycerol and the fatty acid. We want to be able to stimulate the breakdown of these actual triglycerides. So what is that called? When you want to break down lipids into glycerol and fatty acids. If I want to do that, I want to break this down into glycerol and fatty, I'm going to put FA for fatty acids. That process is called lipolysis. What type of receptor would we want there? Generally, we would say, okay, oh, beta 1, right? Well, there's a modified beta 1 receptor. That modified beta 1 receptor is called a beta 3 adrenergic receptor. Thankfully, there isn't many beta 3 adrenergic receptors in the body except for in the adipose tissue, and the recent research has shown also on the detrusor muscle. That makes it a lot easier. So if you can remember, the beta 1 is on the heart and the JG cells of the kidney. The beta 3 is mainly only on the adipose tissue and on the detrusor muscle. Alpha 1 is on the vascular smooth muscle that needs to be stimulated or constricted, and the beta 2 is on the vascular smooth muscle that needs to be inhibited or relaxed. Makes it a lot easier. Okay, let's come over to the pancreas. What about the pancreas? Okay, well, there's two parts of the pancreas, right? You have the acini, which is important for the digestive juices, and then you also have the islets of Langerhans, okay? Now, the islets of Langerhans, if you remember, we had this in an individual video in our endocrinology system. We have two different types of cells, right? Alpha cells and beta cells. The beta cells were the ones that released insulin, and the alpha cells were the ones that released glucagon. And if you remember, insulin was basically trying to increase protein synthesis, increase glycogenesis. It's trying to increase glucose uptake. So it's all generating pathways, anabolic pathways. Glucagon is trying to do catabolic pathways, break down glycogen into glucose, make glucose from non-carbohydrate sources, and break down lipids. Well, that sounds like a good idea in this situation because I want to get as much glucose into the blood and I want to get as much fat into the blood as I possibly can. So if that's the case, I'd want to stimulate this sucker and inhibit that sucker. So guess what? It's a little weird. Okay, the alpha cells, we said that the sympathetic nervous system, we would prefer to stimulate this one. And then the beta cells, we would prefer to inhibit that one. Remember, I told you if you want to inhibit things, usually that's beta 2. So guess what? It's not beta 2 though in this one. It's actually alpha 2 adrenergic receptors. So alpha 2 adrenergic receptors are located on the beta cells, and if these are stimulated by the actual norepinephrine or the epinephrine, it's going to increase potassium ion efflux, which is going to affect the beta cells from being stimulated. So if that's the case, then it's going to inhibit the release of insulin. The alpha cells, though, we want to stimulate them, and so weird as it is, they also have alpha 2 adrenergic receptors. But whenever they're activated, it actually acts on the alpha cells, it stimulates the release of glucagon. And again, glucagon is going to increase your blood glucose levels by gluconeogenesis and glycogenolysis, and it's going to act on your adipose tissue and cause the breakdown of lipids via lipolysis. So that should make sense. Liver. Again, the liver is a super metabolic organ. You should thank your liver for what it does for us. But one of the big things that the liver does is two significant pathways. One is it breaks down glycogen into glucose. And another thing that it can do is it can take amino acids, it can take glycerol, and it can take lactic acid and convert that into glucose, right? And thankfully, we have receptors that are located on our liver that can actually stimulate the glycogen to glucose stimulation and lactic acid, glycerol, and amino acids to glucose. What do you call this bottom one? Lactic acid, glycerol, and amino acids making glucose. They call that gluconeogenesis. We want to increase that activity. We want glucose in the blood so that we can have our muscles having an energy source. We want to break down glycogen into glucose. That is called glycogenolysis. We want to increase that activity. So we need to have receptors that are located on the liver. And these receptors are called beta 2 adrenergic receptors. Okay? Now, let's go to the next one. The next thing that we want to do is we want to focus on the respiratory system. The respiratory system, the biggest thing is we know that we want to get as much nutrients into the blood, fats, glucose, right? We also want to increase the blood flow to the actual skeletal muscles. We want to decrease the blood flow to the non-vital organs. We don't want our GI tract to be functioning, and we don't want our urinary system to be functioning. But we want as much air to come into our lungs as we possibly can because we want to get those lungs properly ventilated so that we can have good perfusion. So if that's the case, then we want to make sure we open up those bronchi as best as we possibly can. So now, in the actual bronchi, so here's our bronchi, and I'm just kind of zooming in on that bronchi, which is going into the lungs, right? There's smooth muscle here within the bronchi. They call it the bronchial smooth muscle. The bronchial smooth muscle is really, really rich in special types of adrenergic receptors. And those are beta 2 adrenergic receptors. Remember, if we want the smooth muscle to relax, we throw on some beta 2. If we want the smooth muscle to contract, we throw on some alpha 1. But we want the smooth muscle to relax. If the smooth muscle relaxes, what happens to the diameter of this airway? It's going to increase, which is going to cause more ventilation. More ventilation increases perfusion. So we're going to have more oxygen in the blood. Another thing is, you know, which is really cool, when I was doing some research here, there was these suckers here called your mast cells. They sit in the lamina propria, right? The connective tissue, the areolar connective tissue. And these mast cells, it's really, really cool. They also have receptors, and these receptors are also beta 2 adrenergic receptors. And whenever epinephrine and norepinephrine acts on these beta 2 adrenergic receptors on the mast cells, it inhibits the mast cells from releasing histamines. If the mast cells don't release a lot of histamines, what's it going to do to the blood flow going through this area? So imagine here I have a little blood vessel. The bronchial, remember these are our bronchial arteries. This is not the pulmonary arteries. I'm going to repeat that one more time. This artery is not a pulmonary artery, it is a bronchial artery. Now, histamines can act on your bronchial arteries, right? And generally what they do is they cause vasodilation and increase the capillary permeability. Well, if there's decreased histamines, are your blood vessels going to dilate? No, instead there's actually going to be more constriction of those bad boys. So what that does is it decreases the blood flow. And if you decrease the blood flow through the bronchial arteries, it's going to decrease the secretions too, because your glands, the different types of seromucous glands, they depend upon the blood flow. If the blood flow is low, you don't have enough blood going to that seromucous glands, you're not going to make a lot of that mucus. So it's going to decrease secretions. Think about that. If you decrease the secretions within the airway, is it going to be as plugged? No. So now you're going to have an open, patent airway for the air to flow through. All of these supporting the sympathetic nervous system. Holy frick. Okay, let's go to the male reproductive system. If we go to the male reproductive system, I want you to remember this. Parasympathetic, point. That's for the erection. Sympathetic is for the shoot, for the ejaculation. So sympathetic should be on all the structures that are going to support the ejaculatory response. What are some of these structures? Remember we had what's called the epididymis. So here we're going to call this the epididymis. Then we're going to have this tube which is going up, and this tube is called the vas deferens. Then we're going to have a vesicle here, which is making specifically different types of seminal fluid, and this is called your seminal vesicles. And then the other one is going to be your prostate gland. All right? All of these structures are extremely important in the ejaculatory response. Now there's smooth muscle, which is located within the epididymis and the vas deferens. So there's some smooth muscle here, all the way up, and then all the way over here. So we want this smooth muscle to contract. If we want it to contract, what type of receptor should we throw on those suckers? Alpha 1. So I want alpha 1 adrenergic receptors here. I'd want the alpha 1 adrenergic receptors on the vas deferens. Also, there's a little bit of smooth muscle around these actual glands, some different types of myoepithelial cells. And whenever they contract, it helps to squeeze some of the actual seminal fluid into this vas deferens. So now we want to stimulate those puppies. So if that's the case, let's have alpha 1 adrenergic receptor, same thing. Alpha 1 adrenergic receptor. And again, what's the whole purpose of this? If this epididymis is contracting, it's pushing the sperm up into the vas deferens. If the vas deferens is contracting, it's pushing the sperm forward throughout the male reproductive tract. If the seminal vesicles are contracting, they're squeezing out the seminal fluid. If the prostate gland is squeezing, it's actually squeezing out the prostatic fluid, and then it's going to help to make the little swimmer. So then out of here is going to become the little swimmer, and he's looking to target a ovary, right? Or target specifically an oocyte. Okay, got that part. Now let's go to the uterus. Okay. For the uterus, it's extremely interesting. The uterus has smooth muscle, right? Within the actual, they call it the myometrium, right? So you know you have the endometrium, myometrium, perimetrium. This red layer here is called the myometrium. Now, the actual smooth muscle on the myometrium can have two different types of adrenergic receptors. One is alpha 1, and the other one is beta 2. Beta 2, though, is the one that's more closer within the actual labor periods, like getting towards the end of labor. Getting towards the end of gestation. Here's why. In a situation where a person has, let's say, premature labor, right? They go into premature labor. If they start going into premature labor, their uterus is trying to contract and expel the baby out before it needs to be expelled out. And we don't want that to happen, right? We don't want this baby to be coming out anytime soon. We want to wait a little bit. So if that's the case, we need to give some type of drug that can bind onto a beta 2 adrenergic receptor while they're pregnant. We need to give them some type of beta 2 adrenergic receptor, some type of drug that can bind onto it while they're pregnant, that it prevents the premature labor contractions. We want to slow these premature labor contractions down. They call this type of drug that you can give a tocolytic agent. Okay, usually they can give like terbutaline. Not really important within this, but I'm just giving the whole purpose here. If norepinephrine and epinephrine bind onto the beta 2 adrenergic receptor, what do we say it does to the smooth muscle? It relaxes it. If it relaxes, it's not going to start splashing out the baby prematurely, okay? Okay. Now, we have one last thing, thank goodness. And that is going to be in the blood. In the blood, we have these tiny little cytoplasmic fragments, which are called platelets. So let's say here I have some platelets. Whenever the sympathetic nervous system releases norepinephrine or epinephrine, these two chemicals can bind onto these receptors that are present on these platelets. And this is why sometimes stress can be a very bad thing. There's receptors located on these platelets, which are called alpha 2 adrenergic receptors. So again, here's one here, and this is going to be alpha 2 adrenergic receptor. Let's say here's another one, and this is an alpha 2 adrenergic receptor. If it acts on these alpha 2 adrenergic receptors on the platelets, guess what this does? It triggers these platelets to start making chemicals. And the chemicals that these platelets start making are very, very dangerous. Some of these chemicals are like thromboxane, A2. Another one is called serotonin. And then the worst one is ADP. If you guys remember from hemostasis, these chemicals, ADP, serotonin, thromboxane A2, these particularly caused a problem, and this situation was called platelet aggregation. And if you increase platelet aggregation, what are you going to increase the formation of? A deadly situation, right? That it could be deadly. And that is going to be the increased formation of possibly a thrombus. Okay? All right, engineers, I hope this video made sense. I hope that you guys did like it. I hope that you learned something. If you guys did, please hit that like button. Comment down in the comment section. Please subscribe. Also, guys, if you get a chance, go check out our Facebook, Instagram, and even our Patreon account. If you have the opportunity to donate, we would truly appreciate it. All right, engineers, as always, until next time.